Hi everyone, it's Mr. Sinti, and today I'd like to talk to you about the structure of DNA, the structure of deoxyribonucleic acid. And it's a fascinating structure. It's one of the most important molecules in biology, and therefore its structure, the way it looks, is going to give us some insight and some clues on its functionality. That is, how it replicates and how it codes for making proteins. And so our discussion, let's jump right into it. Our discussion begins with two individuals by the name of Jim Watson and Francis Crick, and they get credit for discovering the famous double helix structure of DNA, and they did this by building models in their lab room at Cambridge University in England. And they just didn't come up with this on their own. The story that I'm about to share with you is a collaborative. They picked up information along the way in their study from several important scientists pictured here below. Linus Pauling, this is Maurice Wilkins, and Rosalind Franklin. And so individuals, there's another one by the name of Erwin Chargaff. They all kind of contributed little pieces of the puzzle to the structure of DNA. So it's kind of fascinating. I hope you'll enjoy this. And so let's begin our look here. Let me enlarge this and come over here if I can. Click on this. And so I want to start this conversation, in terms of our timeline. We're talking about the early 1950s, because if you remember from our previous discussion that it was just determined from the Hershey-Chase experiment that DNA was a genetic material, because it was the molecule that went inside of bacteria that caused them to replicate viruses. And so now it was a bona fide race, meaning that whoever were the scientists to come up with the structure of DNA were obviously going to become quite famous, and so very important. And so let's take a look at this. Right out of the gate, what I want to say about DNA is that it's a polymer, and it's made up of repeating subunits, and the monomer that it's composed of is something called a nucleotide. And so when you put it together, just as we did with sugars, we said the monomer was a saccharide, so it's a polysaccharide. Or if in a protein, if the monomer was a peptide, a protein would be a polypeptide. So DNA is a polynucleotide. And so what is a nucleotide? If it's just a chain of nucleotides, this is a nucleotide. And an individual nucleotide is actually composed of three smaller subunits. And the three smaller subunits are something called a phosphate group. It's called a phosphate group because there's a phosphorus element, and it's surrounded by oxygen, as you can see here. And so if I were to illustrate this, sometimes we don't like to draw in all the detail, and so we could sort of shorthand this phosphate group. We can draw a single P for the phosphorus, and see how it has like this little halo of oxygen around it? We could simply draw a circle around it this way. You might recall this from a past discussion that this functional group is present in a phospholipid, which is a component of the cell membrane. So what I want to say about a phosphate group is that it's negatively charged. This oxygen has a negative charge to it, and sometimes that hydrogen is able to jump ship, and there's another negative charge over here. So it's a negative functional group. This phosphate is connected to, so that's part one. So one part of a nucleotide is the phosphate. The second part is a sugar. You're like, well, what sugar is it? Is it glucose? Is it fructose? Is it galactose? Is it lactose? No, it is a sugar. It's a 5-carbon sugar. So it's 1, 2, 3, 4, and 5. Sometimes that's labeled, I'm sorry, let me erase that. Sometimes these carbons are labeled around the horn like this. This is carbon 1, 2, 3, 4, and 5. That's going to actually become important to us. Whoops, sorry about that. This happens every once in a while. I will not update that. Let's go back over here. So this is carbon 1, 2, 3, 4, and 5. Sometimes that is important when we talk a little bit more about replication. We're going to talk about how replication occurs in this 3' direction over here. These are prime carbons. So it's a 5-carbon sugar. Sometimes we call this a pentose. For 5-ose is the suffix we put on the name of a sugar. What's it called? It's famously called deoxyribose. Deoxyribose is the name of the sugar. You're like, huh, I wonder why it's not just called ribose. What's up with the deoxy? Well, when something's D, it's like dehydrated, means it's absent, or it's less something. So deoxy, do you notice here on the second carbon, instead of an OH or a hydroxyl group coming off of both carbon 3 and 2, there's no oxygen. Let me draw in the oxygen. If I were to draw in the oxygen right there, then it would simply be the ribose sugar. But it doesn't have the oxygen. It's devoid of the oxygen, and so therefore it's considered to be deoxyribose, because without the oxygen. So here we have a phosphate, a deoxyribose sugar, and now this third component is called a nitrogen base. Why nitrogen? Because in the ring of carbon that makes up this structure, there's nitrogen embedded in it. Do you see how there's 3 nitrogens over here? And so, as it turns out, that there's 3 components of a nucleotide. There's the phosphate, the sugar, and a nitrogen base. As it turns out, there's 4 kinds of nitrogen bases. And so there's going to be actually 4 kinds of nucleotides. And so let's look at this sort of simple cartoon drawing up. Here's a phosphate, here's the sugar. Here's a nitrogenous base, or nitrogen base. Here's a phosphate, sugar, and a nitrogenous base. Do you notice how this base has 1 ring, and this one has 2 rings? So some nucleotides have a single base, and some have a double base. This is a term that may seem sort of trivial or detailed at the moment, but it'll become important as we talk about DNA structure more and more. So a single base is called a pyrimidine, and a double base is called a purine. Let's take a look at what these actually look like. So here are the 4 nucleotides, and they're very famous. You may know them somewhere in your past. So let's see if we can take a look at this. This first nucleotide, and when I say it's a nucleotide, it means that it's composed of a phosphate. Here's the sugar deoxyribose, and a nitrogen base. This particular nitrogen base is a double one, so it's purine. And this nitrogen base is called adenine. And so this whole nucleotide, this whole thing is a nucleotide, we could actually say it's A. We can take just adenine and shorten it to A. Now, why can we do that? Because all 4 of the nucleotides, as you can see, have the same phosphate, sugar, phosphate, sugar, phosphate, sugar. So the only thing different are these nitrogen bases. So one of them is called adenine, one of them is called guanine. It's also a double ring. The other one is called thymine. It's a single ring, or pyrimidine. And the 4th one is cytosine. And so we have these 4 letters that compose the structure of DNA: adenine, guanine, thymine, and cytosine. And so here's a more detailed look at these. Guanine and adenine are the double rings, or purines, and cytosine and thymine are the single rings. So when you start putting these things together, let's sort of link it together, because we said the DNA was a polymer. Let me draw over here on the side. Let's say that, let's go up here. I'll just sort of redraw this along here so you can follow along. If we have a phosphate connected to a sugar, and then we have one of the 4 nitrogen bases, what shall it be? Let me just copy this. Let's say it's thymine. I'll put a T here. So this is my little shorthand drawing of this. Phosphate, sugar, thymine. This would be 1 nucleotide, these 3 components, phosphate, sugar, base. Remember we said the DNA was a polynucleotide, which means that they're connected together like beads. So there's one connected, there's another one connected, there's another one connected. So if I went straight down the line here, I could say that this phosphate, this sugar is going to connect to another nucleotide, which is, I'll just make it adenine, and then we'll come down here, sugar, and let's make this one cytosine. Here's a phosphate group, and we'll make that a guanine. And so if I were, let's change the color, let's circle the nucleotides. There's 1 connected to another one there. Wait, did I mess that up? Phosphate, sugar, base, phosphate, sugar, base. I think I screwed this up. Sorry. That's embarrassing, but it's okay. It'll happen. I can actually make that larger. Let's do this, yeah. Okay, let's go back here and we'll do this. So phosphate, sugar, base. Whoops, too big. Sorry. So phosphate, sugar, base is one nucleotide, and then we'll connect that to a phosphate, sugar, and a base. That's the second one. And then here's another one, phosphate, sugar, base. And then here's the fourth one, phosphate, sugar, and base. So now that I believe that's correct. So here's one nucleotide to the next one, to the next one. So it's 4 of them. Now, the thing about DNA is that it's more than one strand. And so do you see how this is one strand or polynucleotide? There's another strand to it, and sometimes you know the name of DNA is often called the double helix. And so double implies that there's not just one polynucleotide strand, but there's 2 of them. So they have to be held together. Do you notice here how the side of one strand is made up of phosphate, sugar, phosphate, sugar? So it's kind of like a ladder, if you will, and it's alternating nitrogen bases. If you put the other strand together, you can notice that here is the phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base, and they're connected together in the center of this ladder. The nitrogen bases are holding the structure together. And what's holding the nitrogen bases together are these weak interactions called hydrogen bonds. You may recall those from polar molecules. And so these hydrogen bonds are holding the double helix together. And so let me sort of take it off of that for a second. And let's go here if I can. Let me try to get to a black drop right there. There we go. Let's take a look. So if I had something like this, if I had phosphate, sugar, and then I threw in that. And then I asked myself, well, on the other side, phosphate, sugar, I asked myself, what is holding the molecule together? And this was a question that scientists had for a long time. They were trying to figure out what was holding the 2 strands together. Now, if you remember from our picture just a second ago, thymine is the nitrogen base that adenine always binds with, and thymine over here would bond with adenine. So if I threw in another one, if I said that this was guanine over on this side, whoops. If I threw in a guanine here, this means that guanine is always going to bind with cytosine. So this is known as the base pair rules, that these 2 are always bonding with one another through hydrogen bonds. So T binds with A and C binds with G, and these are the number of hydrogen bonds. So how do we know this? What's going on with this? Let's take a little bit of a look at some video that we have here. And so let's take a look first off at these building blocks right here. And so first thing I want to show you is this little video clip right here. Here are the 4 nitrogen bases, adenine, cytosine, guanine, and thymine. And so notice how this is a double ring, and this is the double ring, and these are the 2 single rings. So these are the 4 bases. So we know that these are the bases that comprise the DNA molecule, but the question that scientists had when they were looking at the structure of deoxyribonucleic acid, let me just back that up for a second. So this is what DNA is actually standing for, deoxyribonucleic acid, so it's DNA. It's called deoxyribo, you now know what that means. It's the sugar, deoxyribo, and it's nucleic because DNA was largely found in the nucleus of eukaryotic cells. Acid, because that phosphorus group can donate hydrogens and it has acidic properties. And so I want to talk a little bit about here, let's go here. Actually, let's go here. When you look at the structure and you ask yourself the question, like, how do we know what bases are attached to which? There's a scientist by the name of Erwin Chargaff, and whenever he took DNA from an organism, he could extract the DNA from it, and he was able to, he didn't know the structure, but he was able to tease apart the nitrogen bases in the molecule. And whatever he looked at, it seemed like the amount, see how these are like little tiles here? It seemed like the amount of cytosine and guanine always equaled each other, and adenine and thymine always equaled each other. And let me just let this play. And it didn't matter what organism he was looking at, so he tried different things. Like he would investigate like salmon. He would some salmon, break open the cells, an ox, he would look at bacteria. And every time he extracted the DNA and he counted the amount of these nitrogen bases, let me just sort of pause it at this slide right here. Do you see C and G always equal, A and T always equal, C and G always equal? There's something to this, even though he didn't know the structure. So let me sort of write on top of this. If I told you that, for example, adenine, let's just make this up. Here's a bacteria. Let's consider this right here, the bacteria. Let's say, let me make this up. Let's say the amount of adenine was, now this is obviously out of the four of them, 100%. Let's say adenine is 15%. If adenine is 15%, he always found the amount of thymine also being 15%. You're like, what of that? You're like, well, I'm not sure. So what's cytosine and guanine? Would you be able to figure that out from this conversation? You're like, well, I don't know. It seems like 15 and 15, if you add that up, is 30. So that means that the remaining amount must be 70. So you're like, well, how much would C and G be? That means that the percentage of this would be 35 and 35. It always seemed to work out that way. There must be some relationship between C and G and A and T. It's quite clever, I think, the way he was able to deduce that. So let's go back to the slides and take another look at that. And so along comes this scientist by the name of Rosalind Franklin. And Rosalind Franklin, this is a picture of her. She was working in King's College, which is in London, England. And she was an X-ray crystallographer. And she was working along with Maurice Wilkins. And you know what X-rays, I think some of us are familiar with X-rays when you go in and have your teeth X-rayed, or if you had a bone broken. So these short waves that go in, and you can strike something, and you could see something that would be normally impossible to see. So she was extracting DNA and shooting X-rays at it, and I'm not sure what you would think she would see, but this is the actual photograph. It's a rather famous story. I won't go into the detail of it, but this is her famous photo, photo 51. And do you see how it's this X-shaped pattern right here? To a trained X-ray crystallographer or a biochemist, you can deduce from that molecule, from this picture, that there were two strands in the molecule, and it was helical, meaning that the two strands of the ladder sort of twisted, and I can show you another picture of that later, and that the distance between the ladder was even. And so it was a ladder, in other words, let me sort of redraw that. In other words, this picture was able to show that the ladder was equal in distance. It wasn't but like bulging one more than the other. And the only way that it could be even like this is if a double ring paired up with a single ring. And then a single ring could pair up with a double ring can pair with a single. Do you follow this? So it's like double ring, single ring, double ring, single ring. So the distance, this distance right here was uniform, and it was a two-stranded molecule. So this was a really important contribution to the structure of DNA, and you're like, well, how come Rosalind Franklin didn't receive the Nobel Prize for determining the structure since she took this critical X-ray picture? It's an interesting story. She probably would have, but she actually passed away from ovarian cancer as a result of all of her X-ray work. It's kind of a sad story, but she's certainly very important in this conversation. And so let's go back to this gentleman, Jim Watson, an American scientist who was working with Francis Crick at Cambridge in England. And when Jim Watson and also Francis Crick saw that photograph, it became very clear to them that they were dealing with two strands, and they were dealing with a molecule that was equal in distance from one strand to the other. So they sort of figured that the nitrogen bases ultimately were located on the inside and the phosphates were on the outside. It's an interesting story. I mentioned earlier that there was another person involved by the name of Linus Pauling. Now, Linus Pauling, if you're able to follow all of this, where are we going here? Let's try this one. Linus Pauling was a Nobel Prize winning biochemist working in California at CIT. And everyone thought in the 1950s, gee, Linus Pauling, he's one of the greatest chemists. He's going to be the one that determines the structure of DNA. We probably really shouldn't even be working on it. He's the guy. He's going to be able to do it. And so, as it turns out, he was working on it. And he thought, and he put out a paper, he thought DNA was made up of 3 strands. He thought the phosphates, remember those were negatively charged, were located on the inside. But as it turns out, when you have things that are negatively charged of the same kind, they will repel. And the molecule will become unstable, and it will break open and fracture. And so Linus Pauling published this paper, and the rest of the biological community kind of said, no, that's totally wrong, and Linus is, mistaken. But, this is the thing in science, is like an error leads you to the correct thing. And so it's kind of fine that that happened. And so as it turns out, the real structure of DNA looks a lot like this. Let me just play this and let's talk about it. So you've got these 2 strands, so it's considered to be double. And on the sides of the latter are the sugar phosphates, and in the center of the molecule are these nitrogen bases, which are paired together. And the molecule is complementary, meaning that A bonds with T, C bonds with G, and you can sort of see from that model how the molecule could replicate. And so as it turns out, when you look at this helical, there's kind of a big groove right here. It's called the major groove. And then over here, this one's called the minor groove. And you're like, well, that's kind of a little bit of a detail. But actually, that's going to be important when we talk about how DNA is replicated and how it's transcribed in the future. And so this is kind of a cool animation. When you break it down like this, you could look here in orange at the sugar phosphates. Here's a phosphate, which is negative, connected to the sugar. So the side is phosphate, sugar, phosphate, sugar, phosphate, sugar. So just a little thought as well. I'll just let this continue to play. If the DNA, just keep in mind what DNA is. It's the genetic molecule. And so that it's somehow coding all of our traits. If it's coding all of our traits to determine all of our features, do you think the genetic code, the difference between one organism and another, is phosphate, sugar, phosphate, sugar, phosphate, sugar? Or do you think the uniqueness of the molecule happens to be the sequence of nitrogen bases? So it's significant to know that A, T, C, C, G, T, that actually means something. That's the genetic code. So the sequence of nitrogen bases is rather important when you're talking about the structure of DNA. So this is one strand, and then when you bring in the other strand, it sort of coils along together. Here's one strand over there. So this is one polynucleotide. This is the other polynucleotide. So DNA is really a double polynucleotide. It's held together by nitrogen bases. Here's the purine, here's the pyrimidine, and in this case, this is cytosine, this is guanine, and they're held together by 3 hydrogen bonds. What's funny about hydrogen bonds is that they're kind of weak, but they're strength in numbers. But you don't want the molecule to be perfectly stuck. You want it to be able to peel open so that you can read it. It's kind of like a book that would be useless if you couldn't open it. So the molecule opens rather easily. So you can see here that the bases sort of stack up in the middle. And when you put the whole thing together, it forms this complementariness between A, T, C, and G. And so here we have the double helix. And let me take you back over here to the slides if I can. Let's go here, and then there, and then maybe I'll even go here. And so this is a picture, you might recognize this from our class website. This is a picture of Jim Watson and Francis Crick. And what they did is they pieced together all of this information, Chargaff's base pairing rules, Rosalind Franklin and Maurice Wilkins X-rays, and they just simply created a model in their lab at the Cavendish at Cambridge University. And they began to work on this. And here's another picture of kind of famous Jim and Francis discussing the structure of DNA and how it's made up of a polynucleotide. And here are the nitrogen bases. Here's the deoxyribose sugar. I'm not going to talk to you in this slideshow about uracil and ribose, but those are constituents in RNA. That's coming up in a future video. And then finally, here's the molecule as we know it today. It's a double-stranded, double polynucleotide held together by hydrogen bonds. Here is it forming into the helix right there. And the molecule is it would be too wide if it's double-stranded, and it would be too narrow if it was single molecules, but it's perfect when you have a double and a single pairing together with one another. And so here's a picture of Erwin Chargaff, and he was the one who was breaking up DNA into its components, and he was studying different organisms. And when he was doing that, he was coming up with his sort of base pair rules that A always binds with T and G always binds with C. And he was able to come up with these Chargaff rules and that they are equal to one another. And so just, not that this is testable, but I found that this is kind of curious, is that in humans it's 30% A and T and 20% G and C. That's what a human is composed of. And so here we have a close-up of the hydrogen bonds between A and T and G and C, holding those together. And then here in this picture is Francis Crick. Here is Jim Watson and Maurice Wilkins receiving their Nobel Prize for their great work in 1953 on the structure of DNA. And then lastly, what I wanted to say about this, if I can even get over here, is that I wanted to show you this little video clip of Jim Watson today. Now this was a while ago. Jim Watson is still alive, fortunately. And then this is Watson today. He's obviously a lot older, and so he's telling the story right here. He's sitting back in his old desk at Cambridge University, and he's telling us how when he was trying to build that model of DNA that you saw in the picture, he ordered some molecules of nitrogen bases, but they were slow to make these models. So he said, forget it, I'm just going to cut out some cardboard. And so at his desk, literally, Jim Watson, as he's sitting there, he's taken these pieces of cardboard, and he's like, hey, this is maybe adenine, and this is thymine over there, and he's figuring, look, these are probably where the hydrogen bonds are forming. And so he figured in his mind, well, if that pair was there together, maybe another pair could be stacked on top of that, and another pair could be stacked yet on top of that. And so when he superimposed these base pairs on top of one another, he began to realize something quite magical in the lab. He's like, oh my God, this is how the molecule looks. The sugar phosphates are on the outside and the nitrogen bases are on the inside. And this is a pretty huge molecule. I don't know if you knew this, but there's approximately 3 billion nitrogen bases in human DNA, which means if you took into account like the actual size of the molecule, if you were to stretch it out in one cell, it would be 3 meters long, which is incredible. 3 meters long. That's like 3 baseball bats in length. And all of that fits into the nucleus, which is 5 microns in diameter. So it's like, geez, how do you get all that DNA into a small little nucleus? And you may recall this from before. We talked about it, how DNA can coil itself around histone proteins. And so I hope you enjoyed this conversation about the structure of DNA. It's a great one, and we'll keep coming back to it. And I hope you enjoyed watching that. Thanks.